So from this part, we can see that with this kind of treatment, it will show very consistent B cell depletion in vivo, and also it shows some effect in the treatment of the SLE symptoms. But the therapeutic effect is not ideal, because we found that the autoantibody generation of the mice still remains in the similar level with the treatment of the CAR T cell, and also in the control groups. And also the some of the measures of the kidney function showed that although the deposition of the immune complex has showed less in this treated group, but the kidney is not as healthy as we expect. So we try to find out what is optimization strategy we can use in this kind of treatment. So first of all, we think about that in the clinic treatment, when we treat the patient with early stage of SLE, it will help the patient to remain healthier in the long term. So maybe we can try that to treat the MI or LPR mouse at the early age, and to see whether the treatment in the early age will help the CAR T cell to show better efficacy in the long term. So we tried to test that with the different age of MI or LPR mouse, we found that 8 weeks is the age that the autoantibodies has not accumulated in the peripheral, and the skin remains very healthy in this stage. So we use the mouse at the 8th week, and with the treatment of CAR T cells to see whether it will prevent the SLE symptom in the long term. So from here, we can see that at the age of 13 weeks, when the mouse already appears to be with the very severe symptom of the SLE, the CAR T treated mouse appear to be very healthy on the skin, and also on the proteinuria level. And also at the age of 22 weeks, we also tested the condition of the mouse. We found out that not only the survival rate of the CAR T treated mouse is way better than the control groups, and the skin rash remains to be very healthy in this kind of treated mouse, and the glomerulus is also very healthy. And luckily, we found that the accumulation of the autoantibodies showed also very good signal. Like this, we can see that it is the levels of the autoantibodies, such as dsDNA and anti-nuclear antibodies, is very close to the mouse without disease, and also the kidney function is very good. And second, we also think about maybe we can optimize our treatment strategy with different CAR designs. As we know that in the treatment of leukemia or lymphoma, there are different kind of products of CAR T cells. So some of them are with the signal of CD28, and some of them are with the signal of 4-1BB. And the cancer research has showed that the 4-1BB CAR T cell showed the function of CAR T cell in very long term, and also less exhausted stage of the T cells. So we tried to see whether with the second signal of 4-1BB will help the CAR T cell to treat the mouse better. So for this part, we use the 4-1BB CAR T cells to treat the mouse. And similarly, at the first week after the transfer, we can see that the B cell will decrease very dramatically, and there is no change in the plasma cells, and also the CD4 to CD8 ratio. But 9 weeks later, the B cell percentage is also very low, But from this time, we can see that the plasma cell also decreased, and the CD2 to CD8 ratio has altered, which indicated that the mouse has turned to a better immune environment. And for this part, we also tried to test the syndromes of the CAR T of the SLE has how the syndrome has turned out to be. So here we can see that from the survival rate, the treated group showed a way better survival condition of the with the treatment compared to the control group, and also the smaller spleen has been found, and also the level of autoantibodies has been lower in this kind of group, and also the kidney showed better function, indicated by the UPC ratio and also by the slides of the glomerulus. And also we can see that there is almost no immune complex deposition in this kidney. So for this part, this is a summary, and for the whole story, we have found out the CAR T cell treatment for the SLE mouse is very good, but we also tried to make it better with the different strategies, such as treat the mouse with the early age, or also treat the mouse with the 4-1BB CAR T. But what is more delight to us is that after the publication of our work, last year, at the end of last year, there is one case published in the New England Journal of Medicine, showed that the human SLE, when a human SLE treated with the CAR T cell, the female, very young female patient showed that after two years of treatment, the symptom is totally diminished, and also the B cell has grow back to be into a very healthy stage. And also last week, there is a second paper published in Nature Medicine, indicates that the function of the CAR T therapy appear to be efficient in more SLE patients. So this is my story, and thanks to my team members from Zhejiang University Lulingrong Lab and the Serong Hospital Department of Rheumatology. And also I'd like to thank Professor Lingrong Lu for his patient help and the support in this project. And I'm happy to take any questions. Thank you.
Thank you, Jin, for your amazing presentation, very interesting data. May I, sorry, I think we have maybe, no, I have a couple of questions. 4-1BB helps CAR T cells to persist. CD28 helps T CARs to escape exhaustion. What do you think? What is your opinion about the simultaneously providing of CAR T cells with both signaling system?
Yeah, it is a very interesting question, and a lot of researchers is studying these questions. So from current researches, mostly on the tumor researches, which is more well known in the CAR T therapy, the researchers found out that the signal of CD28 is way stronger than the 4-1BB. And with this kind of signal, the T cell will become more easy to be exhausted. And some of the T cell will be very hard to enter the site of tumor again. In our case, we think maybe the CAR T cell will be not very easy to enter the bone marrow, because in this site, it's not easy for the circulating T cell to enter. So that is one of the reasons maybe why the 4-1BB CAR T cell will show better long-term efficacy in this SLE treatment.
And another question, you said that we observed good effect in the prophylactic scheme. with prophylactic regimen and the introduction of CAR T cells. What did you mean? You propose to use CAR T cells in, for example, persons with genetic predisposition to SLE?
Yeah, that's a very difficult question because currently it's very hard to move this forward because of some ethic problems. We hypothesize that maybe the patients with more severe CAR T cell, with more severe syndrome will be more acceptable to the CAR T therapy because maybe they have no more choices. But very controversially, we find that the treatment with the early stage of the disease will show better results. That is not a very easy question to be solved currently.
Thanks. And we have another question, the last one. Have you tested the condition of the transferred CD8 T cells?
Yeah, we have tried to test it, but actually it's not very easy because as I said, that this kind of mouse will accumulate a lot of T cell in the free flow when the disease happens. And that may be one of the reasons keeps way away to find out the T cell we have injected. So, but the function can remain, as I indicated in the previous slides. The function of the killing still remains in the long term. So we can see maybe some of the CAR T cell remains working, but it may need some triggers like the maybe under the new generation of the B cells or some other special condition in the body.
Thanks again for your very interesting report.
Thank you.
And we are moving to our